Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The selective binding of one molecule by another, a process fundamental to life itself, underpins enzymatic catalysis, signal transduction, and countless other biological phenomena. Donald J. Cram sought to mimic these intricate interactions within the realm of synthetic chemistry, crafting molecules capable of recognizing and binding specific target molecules, much like an enzyme embraces its substrate. This ambition drove his early explorations into what would become known as host-guest chemistry, a field that revolutionized molecular recognition and laid the groundwork for modern supramolecular chemistry. His work, acknowledged with a share of the Nobel Prize in chemistry, began with a deceptively simple question: could one design a molecule with a cavity or pocket precisely tailored to accommodate a specific guest molecule? Cram's initial approach centered on the synthesis of macrocyclic polyethers, compounds now commonly known as crown ethers. These cyclic structures, composed of repeating ethylene oxide units, presented a unique opportunity for molecular design. The ether linkages provided flexibility, allowing the crown ether to adopt various conformations, while the oxygen atoms offered binding sites for positively charged ions or other electron-efficient species. The challenge, however, lay in controlling the size and shape of the macrocycle to achieve selective binding. One of Cram's early successes involved the synthesis of crown ethers designed to bind alkali metal cations. These metals, such as sodium, potassium, and lithium, play crucial roles in biological systems, including nerve impulse transmission and enzyme activity. Cram recognized that the size of the crown ether's cavity could be tuned to match the ionic radius of a specific alkali metal, thereby maximizing the electrostatic interactions between the ether oxygen atoms and the cation. Consider, for instance, the synthesis of dibenzo 18 crown 6. This molecule, featuring an 18-membered ring with 6 oxygen atoms and 2 flanking benzo groups, exhibited a pronounced affinity for potassium ions. The 18-membered ring provided a cavity of appropriate size to accommodate the potassium ion, while the oxygen atoms, acting as Lewis bases, coordinated to the positively charged metal. The benzo groups, while contributing to the molecule's rigidity, also influenced its solubility and overall binding properties. This selectivity arose from the complementary fit between the host and guest, a concept that would become central to Cram's approach. The binding event itself was driven by a combination of electrostatic interactions and the favorable release of solvent molecules from the cation's solvation shell as it entered the crown ether's cavity. Crucially, Cram's work built directly upon Charles Pedersen's serendipitous discovery of crown ethers. Pedersen, while working at DuPont, stumbled upon the synthesis of dibenzo 18 crown 6 during an attempt to prepare a different compound. Recognizing the unusual ability of this molecule to complex with alkali metal ions, Pedersen embarked on a systematic study of crown ether synthesis and binding properties, publishing his findings in a series of groundbreaking papers. Cram, deeply impressed by Pedersen's work, saw the potential to extend these initial observations into a more rational and systematic approach to molecular design. Where Pedersen's initial discovery was largely accidental, Cram sought to develop a set of design principles that would allow chemists to create host molecules with predictable binding affinities and selectivities. Cram's innovation lay in his emphasis on preorganization and complementarity. He understood that the most effective host molecules would be those that were preorganized in a conformation suitable for binding the guest. This meant minimizing the conformational changes required for complex formation, thereby reducing the entropic penalty associated with binding. Furthermore, he recognized the importance of achieving a high degree of complementarity between the host and guest, ensuring that the binding site was not only of the correct size and shape, but also possessed the appropriate array of functional groups to maximize attractive interactions. The impact of Cram's early work on crown ethers extended far beyond the realm of alkali metal binding. It provided a blueprint for the design of host molecules capable of recognizing a wide variety of guest species, including organic ammonium ions, amino acids, and even other organic molecules. By carefully controlling the stereochemistry and functionality of the host molecule, Cram and his co-workers were able to create systems with remarkable selectivity, mimicking the exquisite specificity of enzymes. His approach involved a combination of careful synthetic design, detailed structural analysis using techniques such as X-ray crystallography, and rigorous thermodynamic measurements to quantify binding affinities and selectivities. These studies provided crucial insights into the factors governing molecular recognition and paved the way for the development of more sophisticated host-guest systems. The development of crown ethers and their application in host-guest chemistry represented a paradigm shift in the field of chemistry. It demonstrated the power of rational molecular design to create molecules with specific functions, opening up new avenues for research in areas such as catalysis, separation science, and drug delivery. Cram's work provided a conceptual framework for understanding molecular recognition and inspired a generation of chemists to explore the possibilities of supramolecular chemistry. The stage was set for further innovations, including the development of even more complex and sophisticated host molecules as he continued to refine the principles of preorganization and complementarity. The work of Linus Pauling, particularly his concept of molecular complementarity as exemplified in enzyme-substrate interactions and antibody-antigen binding, served as a direct inspiration for Cram's focus on creating synthetic receptors with tailored binding sites. Pauling's emphasis on the precise fit between interacting molecules, maximizing favorable interactions, and minimizing steric clashes, provided a theoretical foundation for Cram's approach to host-guest chemistry. Cram's early work on crown ethers and host-guest chemistry directly influenced researchers such as Jean-Marie Lehn, who further expanded the field of supramolecular chemistry by developing developing more complex and sophisticated receptor molecules. Lehn, who also shared the Nobel Prize with Cram and Pedersen, built upon Cram's principles of preorganization and complementarity to create systems capable of self-assembly and molecular recognition with unprecedented levels of control. The foundation laid by Cram enabled Lehn to explore dynamic molecular recognition and self-organizing systems, pushing the boundaries of supramolecular chemistry even further. The exploration of host-guest interactions using Cram ethers, initially focused on alkali metal ions, soon expanded to encompass a broader range of guest molecules. Cram's group began to investigate the binding of organic ammonium ions, recognizing their importance in biological systems and their potential as building blocks for more complex supramolecular architectures. This expansion required a refinement of the design principles, taking into account the shape, size, and electronic properties of the guest molecule. The challenge of binding organic ammonium ions presented a new set of considerations. Unlike alkali metal ions, which are spherical and carry a single positive charge, organic ammonium ions are often chiral and possess a more complex charge distribution. This necessitated the design of host molecules with chiral cavities capable of discriminating between different enantiomers of the guest. Cram's group developed a variety of chiral crown ethers, incorporating substituents that created a stereochemically defined binding site. One notable example is the synthesis of chiral crown ethers containing bimethyl units. These units, derived from two fused naphthylene rings, possess axial chirality, meaning that they exist as non-superimposable mirror images due to restricted rotation around the bond connecting the two rings. By incorporating these bimethyl units into the crown ether framework, Cram and his coworkers were able to create chiral cavities capable of selectively binding one enantiomer of an organic ammonium ion over the other. This achievement demonstrated the power of stereochemical control in molecular recognition and opened up new possibilities for asymmetric catalysis and chiral separation. The selectivity of these chiral crown ethers was not merely a matter of steric fit. Cram recognized the importance of other factors, such as hydrogen bonding and pi-pi interactions, in determining the binding affinity and selectivity. By incorporating functional groups capable of forming hydrogen bonds with the ammonium ion or engaging in pi-pi interactions with aromatic substituents on the guest, he was able to fine-tune the binding properties of the host molecule. The impact of Cram's work on chiral recognition extended beyond the realm of organic ammonium ions. It provided a general strategy for designing chiral receptors capable of binding a wide variety of chiral molecules, including amino acids, carbohydrates, and pharmaceuticals. This approach has been widely adopted in the development of chiral catalysts, which are used to synthesize enantiomerically pure compounds with applications in the pharmaceutical, agrochemical, and materials science industries. The detailed structural analysis of these host-guest complexes, often using X-ray crystallography, provided crucial insights into the nature of the binding interactions. These studies revealed the precise geometry of the complex, the distances and angles between the interacting atoms, and the role of solid molecules in stabilizing the complex. This information was invaluable for refining the design principles and developing even more effective host molecules. The thermodynamic measurements of binding affinities and selectivities provided quantitative data that complemented the structural information. By measuring the equilibrium constant for the binding of different guest molecules to the host, Cram and his coworkers were able to determine the free energy change associated with binding, as well as the enthalpy and entropy contributions. This information allowed them to understand the driving forces behind molecular recognition and to optimize the binding properties of the host molecule. The careful combination of synthetic design, structural analysis, and thermodynamic measurements was a hallmark of Cram's approach to host-guest chemistry. It allowed him to develop a deep understanding of the factors governing molecular recognition and to create molecules with remarkable selectivity and function. His early work on crown ethers and their application in chiral recognition laid the foundation for a new era of supramolecular chemistry, one in which chemists could design molecules with specific functions and create systems capable of mimicking the complexity and elegance of biological systems. The legacy of Cram's work extends far beyond the specific examples of crown ethers and chiral recognition. His emphasis on preorganization, complementarity, and stereochemical control has become a guiding principle for the design of a wide variety of supramolecular systems, including catalysts, sensors, and drug delivery vehicles. His contributions have had a profound impact on the field of chemistry and continue to inspire researchers today. The principles of host-guest chemistry, pioneered by Cram, are actively employed in contemporary research, particularly in the development of targeted drug delivery systems. Scientists design host molecules to encapsulate drug molecules and selectively release them at the site of action, improving therapeutic efficacy and minimizing side effects. This direct application demonstrates the enduring relevance of Cram's work to current challenges in medicine and materials science. The field of supramolecular chemistry continues to evolve. Building upon the foundations laid by Cram and his contemporaries, researchers are now exploring even more complex and dynamic systems, including self-assembling structures, molecular machines, and adaptive materials. These advancements are pushing the boundaries of what is possible in chemistry and opening up new opportunities for innovation in a wide range of fields. The evolution of supramolecular chemistry, fueled by Cram's groundbreaking work, has led to the development of sophisticated molecular sensors capable of detecting specific analytes with high sensitivity and selectivity. These sensors, based on host-guest interactions, find applications in environmental monitoring, medical diagnostics, and food safety, demonstrating the practical impact of Cram's fundamental research. Cram's legacy lies not only in his specific discoveries, but also in his approach to scientific inquiry. His emphasis on rational design, rigorous experimentation, and a deep understanding of fundamental principles serves as a model for future generations of chemists. His work stands as a testament to the power of human ingenuity to create molecules with specific functions and to unravel the mysteries of the molecular world. The evolution of supramolecular chemistry, fueled by Cram's groundbreaking work, has led to the development of sophisticated molecular sensors capable of detecting specific analytes with high sensitivity and selectivity. These sensors, based on host-guest interactions, find applications in environmental monitoring, medical diagnostics, and food safety, demonstrating the practical impact of Cram's fundamental research. Cram's legacy resides in his specific discoveries and also in his approach to scientific inquiry. His emphasis on rational design, rigorous experimentation, and a deep understanding of fundamental principles serves as a model for future generations of chemists. His work demonstrates the power of human ingenuity to create molecules with specific functions and to unravel the mysteries of the molecular world. A central theme in Cram's research was the development of what became known as host-guest chemistry. This field explores the formation of complexes between two or more molecules, where one molecule host selectively binds another molecule the guest. This concept, while seemingly simple, has profound implications for understanding molecular recognition, catalysis, and the design of new materials. The intellectual roots of this work can be traced back to Fischer's lock-and-key principle, proposed in the late 19th century to explain enzyme specificity. Fischer envisioned enzymes as having a rigid active site lock that could only accommodate specific substrates, the key with a complementary shape. While Fischer's analogy provided a valuable starting point, it also presented limitations. The original lock-and-key model implied a static interaction, failing to account for the dynamic nature of molecular recognition and the flexibility of both host and guest molecules. Additionally, it did address the energetic considerations involved in binding. Cram recognized these shortcomings and sought to develop a more refined model that would incorporate flexibility and preorganization as key factors in molecular recognition. Cram's concept of preorganization constituted a significant advancement over the classical lock-and-key model. He posited that the most effective host molecules are those that are structurally preorganized for binding. This means that the host molecule exists in a conformation that is already complementary to the guest, even before binding occurs. This preorganization minimizes the entropic cost associated with conformational changes upon binding, leading to enhanced binding affinity and selectivity. This concept of preorganization can be understood through an analogy. Imagine trying to assemble a complex piece of furniture. If all the parts are neatly organized and readily accessible, the assembly process is much faster and easier than if the parts are scattered and require significant manipulation to fit together. Similarly, a preorganized host molecule requires minimal conformational change to bind its guest, resulting in a more favorable binding energy. Cram's strategies for preorganizing host molecules involve the design and synthesis of macrocyclic structures, such as crown ethers. Crown ethers are cyclic pilot ethers that contain a ring of oxygen atoms. The size of the ring and the arrangement of the oxygen atoms determine the size and shape of the cavity within the crown ether. By carefully controlling these parameters, Cram was able to create crown ethers that selectively bind alkali metal ions or organic ammonium ions. For example, he synthesized crown ethers with varying ring sizes to selectively bind different alkali metal ions. Sodium ions, potassium ions, and cesium ions each have different ionic radii. Cram designed crown ethers with cavity sizes that closely matched the ionic radii of these metal ions. The crown ether with a cavity size that best fit the sodium ion exhibited the highest binding affinity for sodium. Similarly, crown ethers were tailored to selectively bind organic ammonium ions with specific steric and electronic properties. The energetic advantages of preorganization are substantial. When a host molecule is not preorganized, it must undergo conformational changes to accommodate the guest. These conformational changes require energy, which reduces the overall binding affinity. A preorganized host, on the other hand, requires minimal conformational change, thereby maximizing the binding energy and leading to a stronger and more selective interaction. This is because the preorganized host already possesses the necessary shape and electronic properties to interact favorably with the guest. Cram's work on host-guest chemistry and preorganization was detailed in a series of highly influential publications. In a seminal paper published in 1974, he described the synthesis and binding properties of crown ethers designed to selectively bind alkali metal ions. This work demonstrated the importance of cavity size and complementarity in determining binding affinity. Another key publication in 1978 focused on the design of chiral crown ethers for the ionoselective recognition of organic ammonium ions. This work highlighted the role of stereochemistry in molecular recognition and opened up new avenues for asymmetric catalysis and separation. Linus Pauling's work on molecular complementarity played a role in influencing Cram's thinking. Pauling's studies on antibody-antigen interactions emphasized the importance of shape complementarity and non-covalent interactions in biological recognition. Cram extended these principles to the design of synthetic host molecules, creating systems that mimic the selectivity and efficiency of biological systems. Pauling's concept of induced fit, where the antibody undergoes a conformational change upon binding to the antigen, also influenced Cram's thinking on the dynamic nature of molecular recognition, leading him to emphasize the importance of preorganization to minimize such induced fit costs. Cram's focus on preorganization had a significant impact on the field of supramolecular chemistry. It provided a framework for understanding and predicting molecular recognition events and inspired the design of new host molecules with tailored binding properties. His work also paved the way for the development of molecular sensors, catalysts, and other functional materials based on host-guest interactions. The ability to design molecules that selectively bind other molecules has opened up new possibilities in areas such as drug delivery, environmental remediation, and materials science. The advancement of this field led to the development of molecular containers capable of encapsulating guest molecules within a confined space. These containers, known as cavitans and carcerans, represent a further extension of Cram's work on host-guest chemistry and preorganization. The emphasis on preorganization, born from Cram's meticulous exploration of molecular recognition, furnished a robust foundation for supramolecular chemistry. It offered a lens through which to understand and anticipate molecular recognition events, spurring the creation of novel host molecules meticulously designed with specific binding affinities. This work served as a springboard for the development of molecular sensors, catalysts, and a diverse array of functional materials predicated on host-guest interactions. The capacity to engineer molecules exhibiting selective binding capabilities unlocked new horizons across various disciplines, including targeted drug delivery, environmental remediation strategies, and advanced materials science. This burgeoning field naturally progressed toward the creation of molecular containers, structures designed to encapsulate guest molecules within defined spatial boundaries. These containers, known as cavitans and carcerans, embody a logical extension of Cram's Cram's foundational work in host-guest chemistry and the principle of preorganization. Cavities represent a class of synthetic host molecules characterized by their bowl-shaped or venera-shaped structures. These structures possess a performed cavity capable of accommodating guest molecules of appropriate size and shape. The synthesis of cavities typically involves a multi-step process, often employing cyclization reactions to create the rigid, three-dimensional framework. Early cavities were often based on calixarenes, cyclic oligomers formed from phenyl and formaldehyde units. These calixarenes could be further functionalized to enhance their binding affinity for specific guest molecules. Cram's research group developed sophisticated synthetic strategies to introduce substituents at specific positions on the calixarene framework, allowing for precise control over the shape and size of the cavity. A key aspect of this synthetic approach involved the use of protecting groups to selectively block certain reactive sites during the synthesis, ensuring that the desired functional groups were introduced at the correct locations. For example, tert-butyl groups were frequently employed as protecting groups for the phenolic hydroxyl groups, allowing for selective alkylation or of other positions on the calixarene. The protection could then be achieved under mild conditions to reveal the free hydroxyl groups. One specific example of a cavitan synthesized by Cram's group is a calixarene-based molecule with 4 appended urea groups. These urea groups form hydrogen bonds with guest molecules, enhancing the binding affinity of the cavitan for anions such as chloride or phosphate. The binding of these anions within the cavitan cavity can be detected by various spectroscopic techniques such as nuclear magnetic resonance spectroscopy, providing a means to sense the presence of these ions in solution. This work has implications for the development of sensors for environmental monitoring and medical diagnostics. Carcerands, taking the concept of molecular encapsulation a step further, are host molecules designed to completely imprison guest molecules within their interior cavity. The name carcerant is derived from the Latin word carcer, meaning prison. Unlike cavitans, which possess an opening through which guest molecules can enter and exit, carcerands are typically synthesized as 2 hemispherical fragments that are then covalently linked together, trapping the guest molecule inside. The synthesis of carcerands presents a formidable synthetic challenge, requiring the creation of large, complex molecules with precisely defined shapes and sizes. Cram's group pioneered the development of several elegant synthetic strategies for the construction of carcerands, often employing a combination of convergent and divergent synthetic approaches. In a convergent approach, smaller building blocks are synthesized separately and then joined together in a final cyclization step. A divergent approach involves the stepwise elaboration of a core structure, adding functional groups and extending the molecular framework in a controlled manner. A notable example of a carcerand synthesized by Cram's group is a molecule composed of 2 calixarene-based hemispheres linked by 4 methylene bridges. This carcerand has a cavity large enough to encapsulate small organic molecules such as methane or benzene. Once the guest molecule is trapped inside the carcerand, it is effectively isolated from the external environment. This encapsulation can have a profound effect on the properties of the guest molecule, altering its reactivity, stability, and spectroscopic characteristics. For instance, Cram's group demonstrated that encapsulating highly reactive species within a carcerand can stabilize them, allowing for their study under conditions where they would normally decompose rapidly. This approach has been used to investigate the properties of highly strained or unstable molecules, providing valuable insights into their structure and reactivity. The development of cavitans and carcerands was driven by a desire to create molecular containers with specific functions, such as catalysis or drug delivery. Cram envisioned using these containers to create artificial enzymes that could catalyze chemical reactions with high selectivity and efficiency. The idea was to design cavitans or carcerands with binding sites tailored to recognize specific substrates, bringing them together in close proximity within the cavity and facilitating the desired reaction. While the early attempts to create such artificial enzymes were met with limited success, they laid the groundwork for future research in this area. The concept of using molecular containers to control chemical reactivity remains an active area of investigation, with potential applications in areas such as organic synthesis and materials science. Another potential application of cavitans and carcerands is in drug delivery. By encapsulating drug molecules within these containers, it may be possible to protect them from degradation or premature release in the body, ensuring that they reach their intended target in sufficient concentration. Furthermore, cavitans and carcerands can be functionalized with targeting groups that selectively bind to specific cells or tissues, allowing for targeted drug delivery. This approach has the potential to improve the efficacy and safety of drug treatments, minimizing side effects and maximizing therapeutic benefits. While the development of cavitan and carcerand-based drug delivery systems is still in its early stages, it represents a promising area of research with the potential to revolutionize the treatment of various diseases. Cram's early work in host-guest chemistry, particularly his emphasis on preorganization, profoundly influenced the development of cavitans and carcerands. The principle of preorganization dictated that the host molecule should possess a performed cavity with a shape and size complementary to the guest molecule. This preorganization minimizes the entropic cost associated with binding, leading to stronger and more selective interactions. In the design of cavitans and carcerands, Cram meticulously considered the shape, size, and functionality of the cavity to ensure optimal binding of the target guest molecule. He also recognized the importance of rigidity in the host molecule, as this prevents the cavity from collapsing or destroying upon binding. The synthetic strategies employed by Cram's group were carefully designed to create rigid, preorganized structures with precisely defined cavities. Furthermore, Cram's work on crown ethers provided valuable insights into the factors that govern molecular recognition. He recognized that the binding affinity of a host molecule for a guest molecule depends not only on the shape and size complementarity, but also on the nature of the intermolecular forces involved. These forces can include hydrogen bonding, electrostatic interactions, van der Waals forces, and hydrophobic interactions. In the design of cavities and carcerands, Cram carefully considered these factors, incorporating functional groups that could engage in specific interactions with the guest molecule. For example, he often used urea groups to form hydrogen bonds with anions or hydrophobic groups to promote binding of nonpolar molecules. Cram's contributions to the field of supramolecular chemistry extended far beyond the synthesis of cavities and carcerands. His work established a conceptual framework for understanding and controlling molecular recognition events, inspiring countless researchers to explore the design and synthesis of new host molecules with tailored binding properties. His emphasis on preorganization revolutionized the field, providing a powerful tool for predicting and optimizing molecular interactions. The impact of his work is evident in the development of a wide range of supramolecular systems, including molecular sensors, catalysts, and materials. Researchers such as Jean-Marie Lehn, a co-recipient of the Nobel Prize in Chemistry with Cram and Charles Pedersen, were directly influenced by Cram's work on preorganization and host-guest chemistry. Lehn extended these concepts to the development of self-assembling systems, where molecules spontaneously organize themselves into complex structures. This work built upon Cram's emphasis on the importance of molecular recognition and complementarity in driving supramolecular assembly. Similarly, Fraser Stoddart, another prominent figure in supramolecular chemistry, was influenced by Cram's work on cavities and carcerands. Stoddart developed mechanically interlocked molecules such as rhodoxanes and catenanes, where molecular components are linked together by mechanical bonds rather than covalent bonds. These molecules exhibit unique properties and have potential applications in areas such as molecular machines and nanoscale devices. The design of these mechanically interlocked molecules relies heavily on the principles of molecular recognition and complementarity, which were central to Cram's work. The legacy of Donald Cram's work on cavities and carcerands continues to inspire researchers today. These molecular containers represent a powerful tool for controlling chemical reactivity, stabilizing reactive species, and delivering drugs to specific targets. The principles of preorganization and molecular recognition, which were central to Cram's work, remain fundamental concepts in supramolecular chemistry. The field continues to evolve, with new host molecules and supramolecular systems being developed at an ever-increasing pace. The potential applications of these systems are vast, ranging from medicine and materials science to environmental remediation and energy storage. Cram's pioneering work laid the foundation for this exciting and rapidly expanding field, shaping its direction and inspiring generations of scientists to explore the fascinating world of molecules and their interactions. The creation of molecular containers capable of encapsulating and manipulating guest molecules represented a significant stride toward mimicking the exquisite selectivity and control observed in biological systems. This pursuit of biomimicry extended to Donald Cram's investigations into stereochemical reaction cycles. A domain where the precise spatial arrangement of atoms dictates the outcome of chemical transformations. The challenge lay in achieving stereoselectivity, the preferential formation of one stereoisomer over others in chemical reactions. Cram's innovative approach involved the design of stereochemical reaction cycles, ingenious sequences of reactions designed to amplify and control stereochemical information. His methodology often incorporated chiral auxiliaries, temporary stereogenic units introduced into a molecule to direct the stereochemical course of a reaction. These auxiliaries, carefully chosen and strategically placed, interact with the reactants, creating a steric or electronic environment that favors the formation of a specific stereoisomer. Following the desired transformation, the chiral auxiliary is removed, leaving behind the enantiomerically enriched product. Cram's genius lay in the design of these auxiliaries and the orchestration of the reaction conditions to maximize stereoselectivity. One prominent example of Cram's stereochemical reaction cycles involves the use of chiral crown ethers as auxiliaries in asymmetric synthesis. While his earlier work focused on their host-guest properties, he astutely recognized their potential for stereochemical control. By attaching a reactive functional group to a chiral crown ether, he created a system where the crown ether's chirality influenced the approach of a reactant to the reactive site. The crown ether acted as a stereochemical control element, dictating which face of the reactant was more accessible, leading to the preferential formation of one stereoisomer. Consider, for instance, the reduction of a ketone using a chiral crown ether borohydride reagent. The chiral environment created by the crown ether favors the approach of the hydride to one face of the ketone, leading to the formation of a specific alcohol enantiomer in excess. The stereochemical outcome is determined by the interplay of steric and electronic factors within the crown ether cavity, a testament to Cram's understanding of molecular architecture and its influence on reactivity. The mechanism involves the complexation of the borohydride reagent within the crown ether cavity, followed by the selected transfer of hydride to the ketone substrate. The chiral environment of the crown ether dictates the preferred trajectory of the hydride, leading to the observed stereoselectivity. Another example involves the use of chiral oxazolidinons as auxiliaries in aldol reactions. Cram and his coworkers developed a range of chiral oxazolidinons that, when attached to a carbonyl compound, effectively control the stereochemical outcome of the aldol addition. The oxazolidinon ring creates a rigid framework that shields one face of the carbonyl group, directing the incoming electrophile to the opposite face. This approach allowed for the predictable synthesis of syn or anti-aldol products with high diastereoselectivity. The stereochemical outcome is governed by the Falcon N model, which predicts that the incoming nucleophile will attack the carbonyl group from the face that minimizes steric interactions with the largest substituent on the adjacent stereocenter. These stereochemical reaction cycles found widespread application in asymmetric synthesis, enabling the creation of chiral molecules with high enantiomeric purity. Chiral molecules are essential building blocks in pharmaceutical, agrochemical, and materials science, where their specific stereochemistry dictates their biological activity or material properties. Cram's work provided chemists with powerful tools to access these chiral building blocks in a controlled and efficient manner. The impact of Cram's work extended far beyond the specific examples of stereochemical reaction cycles he developed. His contributions significantly influenced the development of asymmetric catalysis, a field that revolutionized chemical synthesis. Asymmetric catalysis involves the use of chiral catalysts to promote chemical reactions with high stereoselectivity. Cram's emphasis on molecular recognition and preorganization paved the way for the design of more effective and selective chiral catalysts. Reggie Maiori, a Nobel laureate in chemistry, was directly influenced by Cram's work on molecular recognition and stereochemical control. Laureate developed highly efficient chiral catalysts for hydrogenation reactions, enabling the synthesis of a wide range of chiral compounds with exceptional enantiomeric purity. His ruthenium-based catalysts, featuring chiral phosphine ligands, are widely used in industry for the production of pharmaceuticals and fine chemicals. Laureate himself acknowledged the profound influence of Cram's work on his own research, citing Cram's emphasis on molecular architecture and the importance of understanding the interplay of steric and electronic factors in achieving stereo control. Barry Sharpless, another Nobel laureate in chemistry, also drew inspiration from Cram's work. Sharpless developed highly selective oxidation reactions, including the Sharpless epoxidation and the Sharpless dihydroxylation, which employ chiral titanium catalysts to introduce oxygen atoms stereoselectively into organic molecules. These reactions have become indispensable tools in organic synthesis, enabling the efficient synthesis of complex chiral molecules. Sharpless acknowledged Cram's pioneering contributions to supramolecular chemistry and molecular recognition as foundational to his own development of chiral catalysts. Dieta Seabar, a prominent organic chemist, also applied Cram's principles in his development of self-replicating molecules. Seabar's work demonstrated how molecular recognition and stereochemical control could be harnessed to create systems that self-assemble and replicate, mimicking the fundamental processes of life. Cram's approach to stereochemistry was deeply intertwined with his earlier work in host-guest chemistry. The principles of molecular recognition, complementarity, and preorganization, which he developed in the context of crown ethers and cavitans, were directly applicable to the design of stereochemical reaction cycles. He envisioned the reaction site as a host that selectively binds and orients the reactant's guests, dictating the stereochemical outcome of the reaction. This host-guest perspective provided a powerful conceptual framework for understanding and controlling stereoselectivity. The design of chiral auxiliaries, in particular, benefited greatly from Cram's insights into host-guest interactions. He recognized that the effectiveness of a chiral auxiliary depended on its ability to selectively bind to the reactants and create a well-defined stereochemical environment. The better the auxiliary recognized and interacted with the reactants, the higher the stereoselectivity of the reaction. Cram's legacy in stereochemistry lies in his ability to bridge the gap between supramolecular chemistry and organic synthesis. He demonstrated how the principles of molecular recognition and preorganization could be applied to control the stereochemical course of chemical reactions, leading to the development of powerful tools for asymmetric synthesis. His work not only advanced the field of stereochemistry, but also paved the way for the development of asymmetric catalysis, a field that continues to transform the way chemists synthesize chiral molecules. His emphasis on understanding the interplay of steric and electronic factors in achieving stereo control remains a guiding principle for chemists working in this area. Donald Cram's profound contributions irrevocably reshaped the landscape of supramolecular chemistry, establishing molecular recognition as a cornerstone principle in advancing stereochemistry through innovative design. His work created a paradigm shift, moving beyond the synthesis of the individual molecules toward the construction of complex, functional systems held together by non-covalent interactions. The development of crown ethers stands as a testament to Cram's ingenuity. These cyclic polyethers, capable of selectively binding metal cations, provided the first tangible examples of host-guest chemistry. Cram's systematic exploration of crown ether structures and their binding affinities revealed the importance of size, complementarity, and preorganization in molecular recognition. He meticulously crafted crown ethers with varying ring sizes and substituent groups, demonstrating how subtle structural modifications could dramatically alter their selectivity for different metal ions. This work provided a blueprint for designing molecules with specific binding properties, a concept that has since been applied to a wide range of applications, from ion sensing to drug delivery. The impact of crown ethers extends beyond their direct applications. They served as inspiration for countless researchers seeking to create molecules with tailored binding capabilities. Jean-Marie Lehn, for instance, directly built upon Cram's work with crown ethers to develop cryptans, more complex three-dimensional macrocyclic receptors with enhanced binding affinities. Cram's exploration of host-guest chemistry extended beyond crown ethers to encompass a broader range of molecular architectures. He introduced the concept of spherical recognition, where a host molecule with a three-dimensional cavity selectively binds a spherical guest molecule. This concept led to the development of cavitans and carcerans, sophisticated host molecules designed to encapsulate guest molecules within their cavities. Cavitans, with their open-ended cavities, provide a binding site for smaller molecules, while carcerans completely enclose the guest molecule, trapping it within a molecular cage. The synthesis of carcerans represented a tour de force of synthetic chemistry, requiring the creation of complex molecular architectures with precisely defined cavities. These molecules demonstrated the ultimate level of control over molecular encapsulation, offering a unique platform for studying the properties of molecules in confined spaces. Researchers like Julius Rebecca Jr. were directly influenced by Cram's work on cavitans and carcerans, developing their own families of self-assembling capsules with applications in catalysis and drug delivery. A particularly elegant aspect of Cram's research involved the design and synthesis of chiral host molecules capable of enantioselectively binding chiral guest molecules. This work demonstrated the power of supramolecular interactions in chiral recognition, paving the way for the development of chiral sensors and catalysts. Cram recognized that the effectiveness of a chiral host depended on its ability to create a well-defined stereochemical environment around the binding site. By carefully controlling the shape and functionality of the host molecule, he was able to achieve remarkable levels of enantioselectivity in guest binding. This approach has been widely adopted in the design of chiral catalysts, where the stereochemical environment of the catalyst determines the enantiomeric excess of the product. Cram's contributions to stereochemistry extended beyond host-guest chemistry to encompass the study of stereochemical reaction cycles. He investigated how the stereochemical outcome of a reaction could be influenced by the presence of chiral auxiliaries or catalysts. His work revealed the importance of understanding the transition states of chemical reactions and how they are affected by the surrounding molecular environment. Cram's rule of stereochemistry, which predicts the major product of certain carbonyl addition reactions based on the steric bulk of substituents, became a cornerstone of organic synthesis. This rule provided a simple yet powerful tool for predicting and controlling the stereochemical outcome of reactions, guiding synthetic chemists in the design of efficient and stereoselective synthetic routes. David Evans, for example, significantly expanded upon Cram's rule, developing more sophisticated models for predicting stereochemical outcomes in a wider range of reactions. The 1987 Nobel Prize in Chemistry, shared with Jean-Marie Lehn and Charles Pedersen, served as a resounding affirmation of Cram's groundbreaking contributions to supramolecular chemistry. The Nobel Committee recognized the profound impact of their work on the development of molecules with highly selective structure-specific interactions. This award not only recognized Cram's individual achievements, but also elevated the field of supramolecular chemistry to a new level of prominence, inspiring a generation of scientists to explore the possibilities of molecular self-assembly and molecular recognition. The influence of Cram's work extends far beyond the confines of academic chemistry laboratories. His principles of molecular recognition and preorganization have found applications in a wide range of fields, including materials science, drug delivery, and catalysis. In materials science, Cram's concepts have been used to design self-assembling materials with tailored properties. For example, researchers have created supramolecular polymers that can respond to external stimuli, such as light or temperature, changing their properties in a controlled manner. In drug delivery, Cram's host-guest chemistry has been used to encapsulate drugs within carrier molecules, protecting them from degradation and delivering them specifically to target cells. This approach has the potential to revolutionize the treatment of diseases, allowing for more effective and targeted therapies. In catalysis, Cram's principles have been used to design highly efficient and selective catalysts for a variety of chemical reactions. Supramolecular catalysts, which utilize non-covalent interactions to bind and activate substrates, offer a powerful alternative to traditional metal-based catalysts. While Cram's work has had a transformative impact on chemistry, it is important to acknowledge some limitations. The synthesis of complex supramolecular architectures can be challenging and time-consuming, requiring sophisticated synthetic techniques. Furthermore, the stability and functionality of supramolecular assemblies can be sensitive to environmental factors, such as temperature and solvent. Despite these challenges, the field of supramolecular chemistry continues to advance rapidly, with researchers developing new and innovative strategies for creating functional molecular systems. Cram's enduring legacy lies in his ability to bridge the gap between fundamental chemical principles and real-world applications. He demonstrated how a deep understanding of molecular structure and interactions can be used to design molecules with specific functions, paving the way for the development of new technologies and therapies. His emphasis on rational design and systematic experimentation remains a guiding principle for chemists working in the field of supramolecular chemistry. Individuals like Fraser Stoddart, who developed mechanically interlocked molecules MIMs such as rhodexanes and catenanes, directly built upon Cram's foundations in supramolecular chemistry, creating molecular machines with potential applications in nanotechnology. Donald Cram's place in the history of chemistry is secure. He stands as a visionary scientist who transformed the way chemists think about molecules and their interactions. His work not only advanced the field of supramolecular chemistry, but also inspired countless researchers to explore the possibilities of molecular design and self-assembly. His legacy will continue to shape the future of chemistry for generations to come.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.